Hello students, today we study in vitro fertilization. We study chapter sexual reproduction in human. Our today's topic in vitro fertilization. In vitro means literally in glass, or in other words the fertilization is allowed to take place in laboratory glass in tubes. The fertilization is done in tubes. Egg and sperm are fertilized in tubes in glass. Hence the term test tube babies. So we called in vitro fertilization with another term known as test tube babies. This technique may be employed where surgery cannot be used to repair blocked oviduct. In previous lectures we study or we see that we can operate to repair the blocked oviduct, but if the case is not supported the repair of blocked oviduct, we have to do in vitro fertilization. In vitro fertilization has received considerable publicity since the first test tube babies were born in 1978. This in vitro fertilization or IV method is very famous among people, and the first baby was born in 1978. The woman given by fertility drugs. In this procedure, the woman may go through various fertility drugs, various hormones to support her pregnancy, which cause her ovaries to release several mature ova simultaneously. The women which can't have babies have a major problem that their ovaries can't produce mature ova, so these drugs are used that the woman's ovaries release several mature ova simultaneously. These ova are then collected by laparoscopy. It is a method in which we suck the ovum from the oviduct. They are sucked up in the fine tube inserted through the abdominal wall. The ova are then mixed with the husband's seminal fluid. We took a glass tube, we put semen and the egg in it and watch under the microscope to if the cell division take place. If the sperm fertilize the ova and the ova become a zygote and it further develop into embryo by cell division. We see cell division under a microscope. Such an in vitro fertilized ovum. One or more of the dividing zygote are then introduced to the woman uterus by means of a tube inserted through the cervix. After the mixing of the semen and the egg, one or more dividing zygote are then introduced in the woman uterus. Doctors simply put the zygote in the woman uterus by means of a tube inserted through the cervix. They put the zygote by a tube which is inserted through the cervix. Usually only one or none of the zygote develop. We put or we insert one or more zygote. They may be two or three, but only one or no zygote develop. Though occasionally there are multiple births, but sometimes there is a chances that two or more zygote develop and give multiple births. The success rate of in vitro fertilization in between 12 and 40%. It is a quite less, 12% or maybe 40%, depending on how many embryos are transplanted. However, new research using time-lapse photography of the developing IVF embryos during the first few days of life could raise the success rate up to 78%. There is a new technique in which the researchers or doctors see time-lapse photography of developing IVF embryo, which increase the success ratio to 78%. It could also reduce the cost from between £5,000 and £10,000 for each treatment cycle. Suppose if we have in vitro IVF procedure one time, we have to pay 5,000 to 10,000 pound at one treatment cycle. If our zygote don't develop into embryo, we have to repeat our cycle to 750 in Britain. The photographs are used to select the best embryo based on their early development. Scientists use the time lapse when the sperm fertilize the egg and these fertilized egg develop into zygote. Scientists saw multiple zygotes and which zygote is developed faster and which zygote is healthier. They choose the healthier and the faster developing zygote and insert it into the uterus of the female. this embryo or this zygote have the higher chances of the embryo fetus development and gave birth to a healthy baby. Thanks for today's lecture. Like, share, subscribe our channel if you like my video.